the next few hours that we have, I would like to apply most of the approaches that Garrett has presented this morning to the specific question of drug addiction, with the goal to tell you a little bit on how far the field now is. So with the goal to give you an overview of the effort towards establishing a circuit model. So the 3 hours are going to be split into 3 parts. We have a first part where I'm going to tell you a little bit how we got started on this question. Then we have a second part where we will review together the paper that you hopefully all read. And the third part, if time still allows, but we can also do this another day, is to give you some ideas on how we would like to continue that line of research towards a more translational approach. Okay, so but just as a starting point, we would distinguish between addiction and dependence. So as we said yesterday, for us, addiction is the compulsive use despite negative consequences, and that contrasts with dependence that is simply defined by the occurrence of a withdrawal syndrome.
I'm sorry. Yes.
By the occurrence of a withdrawal syndrome when you stop using the drug. And these two things are not necessarily the same. Obviously, there also is a withdrawal involved in addiction, but clearly there are different things. To start, we have to remind ourselves that addiction is not a disease that emerges just like that. You have typically an initiation phase where a first exposure of a drug is done typically in a specific context. That then is followed by a phase where you can actually use the addictive substance in a controlled fashion. And that is associated with a pleasure. So sometimes people call this sort of the honeymoon phase of drug addiction. But obviously, when we turn into the compulsive use despite negative consequences, we have this automatic use that may be totally dissociated from any pleasure. We know that while it is difficult, some can actually withdraw, they become abstinent, but we also know that even after prolonged periods of times of abstinence, there is a risk for relapse. So obviously, 2 essential questions, if you're interested in the neurobiology of drug addiction, is what happens between the control consumption and the compulsive consumption? What substrates are responsible for this transition? And likewise, we would like to understand how is relapse generated. So what is going on in the brain of someone when they have relapse? And so we've been working on these questions for now about 15 years. And on the next slide, I have a very simple version of our hypothesis that has sort of guided our experimental work over all these years. And this hypothesis goes something like that. So we believe that everything starts with the convergence of these pharmacological substances on a specific brain system. And that system would be the mesolimbic dopamine system. So here you have a sagittal section of a mouse brain, and you see that you have the ventral tegmental area at the tip of the brainstem that projects to the accumbens and prefrontal cortex. And you already, schematically, we have indicated that there is a inhibitory control of these projection neurons that release the dopamine. So once that dopamine would increase massively because of the pharmacological power of these substances, as a next step, we would induce certain forms of synaptic plasticity. We and others have called these drug-evoked synaptic plasticities. And that would then lead to a remodeling of the circuit function, which eventually drives the drug-adaptive behavior. So this is a model that is quite different from a model of, for example, neurotoxicity, right? And many years ago when I started, some of the initial models were that these substances are neurotoxic. they kill neurons, and that is why you lose control. So this is a different way to see this. And obviously, this hypothesis needs experimental validation. You need, for example, to start to explain why or whether this is indeed true, that these substances converge on that system, and it is through their that effect that you have this cascade that is triggered. This will not be a trivial thing because by the fact that there are pharmacological substances, they have a lot of other effects. So can we sort of extract a common effect on all of these addictive substances? And then, so that certainly kept us busy for a few years. And then starting in the early 2000s, some reports, and we will review them together, made that claim of drug-evoked synaptic plasticity. So if you want to go that route, you actually have to identify these synapses that undergo the change, examine the induction product, the mechanism through which the induction occurs, and also describe the expression mechanism. So we've spent some time looking at that. And it's only recently, and thanks to the techniques that were so nicely described by Garrett, that we now start to understand a little bit this circuit remodeling and try to establish experiments that would allow to establish links of causality between the circuit remodeling and the drug adaptive behavior. So nowadays, the majority of experiments in our lab work something like that. So we have a drug adaptive behavior that we're interested in. That could be relapse, could be behavioral sensitization, could be anything. We then go in and use what we call an ex vivo approach. That is, in an animal that shows that behavior, we prepare slices of the brain. We kill the animal, we prepare the slices, and we look with identified synapses what happens exactly, what are the plasticity mechanisms. This is not only an academic exercise, because what we would like to extract from that is a good idea on how then to be able to manipulate and reverse and restore normal transmission in vitro. Once we have established this based on our knowledge how this plasticity is expressed, we can then apply that in vivo and see whether we have an effect on the drug adaptive behavior. So this is sort of the typical project that when we do this in our lab. So we have obviously first to start with that question of the convergence, and here you have a quite old, hence not exhaustive list of addictive drugs that was proposed to be listed in increasing relative risk to actually induce the disease. So that would start with the hallucinogens, LSD, that have a relative risk of 1, that is, they actually are not inducing addiction, would go all the way to cocaine and amphetamines, which have the strongest risk of inducing drug addiction. So which drugs are missing here? No. Sorry? Yes, so ecstasy, for example, is not on that list because in the 1990s that was not a very... So this is sort of, this is a personal estimation of 2 eminent pharmacologists that said this is about what we think is indeed true. But obviously it's not so easy to actually establish this. And probably the study that came closest of really establishing sort of demonstrating that risk of turning from the controlled recreational use into the compulsive addictive use is this landmark study from Wagner and Anthony, where admittedly in a retrospective fashion, they looked at the fraction of people who would fulfill the criteria of drug addiction after many years of use of cocaine. And as you can see here, after the first year, it's only 5% that seem to fulfill that experiment. This is a little surprising, right? If you think that you would have access to cocaine for an entire year, chances that you get addicted are actually very low. This then, after 10 years, reaches about 15%, and after 20 years, stabilizes below 20%. So for another drug that you might be more familiar with, cannabis, this same curve is more shallow and stabilizes below 10%. So this is sort of an idea of how you can estimate the relative risk of drug addiction as proposed initially. So clearly, this is how these data were published, but you could also redraw the whole thing in a y-axis from 0 to 100, just to really make the point that even with the most addictive substances, such as cocaine, the overwhelming majority can use them for years recreationally without ever getting addicted. So a first message of today, obviously, is not that you now should go and use cocaine because it's relatively safe, but that we have something called the individual vulnerability. So some people are vulnerable to drugs and some are not. So that's a basic question in the addiction field. How can we explain this at the level of the functioning of the brain?
Can I ask a question?
Yeah, sure.
How is that reflected in studies with animals, with rodents?
We're going to get there, exactly. As you will see, there are different steps also in animals. There are some early forms of drug adaptive behavior that we see in every animal, right? So like reinforcement to cocaine is essentially seen in every mouse or every rat. But when we get to the later stages, we see that dichotomy between those that resist to punishment and those that
And is it at the rates like humans?
Well,
10-15%?
Wait, we'll get there. We will show you that it's not always like humans, but it is similar. Okay, so, just for this is taped, so I have to have the disclaimer that obviously cocaine is a very dangerous substance. You shouldn't take it because it increases your blood pressure and gives you stroke and subarachnoid hemorrhage and myocardial infarction and all these horrible things. But of course, we have to know that initially it is relatively safe. You are not going to get addicted immediately. And that is a reality that sometimes is forgotten. So how is it that these different substances might actually converge onto the mesolimbic dopamine system? So this is, as I said, not a trivial thing because chemically these substances are very diverse. And each substance has its own receptor or its own substrate where they bind. And through that, it's not obviously clear why they should have a common effect. So this is the sort of a mechanistic classification that we have proposed together with Mark Angles, where we take into account the very simple circuit diagram of the VTA, neglecting many things, but this is what we think is sort of the minimally required computational unit to explain the effect of drugs. And you can see that, for example, nicotine, by the fact that the nicotinic receptors are expressed on the dopamine neurons, can directly activate those neurons, cause release, and that would be the way how nicotine increases dopamine release. We also have another group of addictive substances, sometimes also called the psychostimulants, and their primary target is the dopamine transporter. The idea here being that dopamine is sort of an expensive molecule for the brain to produce, which is why there is a very efficient recycling system in place. And that recycling system works at the terminal, where the bulk of the dopamine is being released, but it also works at the level of the dendrites because the dopamine neurons have this capability of releasing dopamine also from their dendrites. And so the dendrites themselves take them up, and when that uptake is inhibited, you have a massive increase of dopamine, and that would be sufficient to drive the initial stages of drug addiction. Okay, so that obviously was challenged experimentally by a famous study in 1998. Because the way they did this, which at the time was sort of the standard in the field, is to create a knockout mouse for the dopamine transporter. And the prediction was, you take away the major molecular target of cocaine, and if it's really through that target that cocaine has its reinforcing effect, you should lose it. That seemed pretty reasonable to me. And when we read that paper, I was a postdoc in San Francisco, there was a big discussion because the observation was just the contrary. So here you have individual mice, the way they learn to self-administer cocaine, and you see that this is the wild type and this is the dopamine transporter, knockouts. So you see that clearly they still self-administer cocaine. Well, maybe not as efficiently as the dopamine, but of course the message from the paper was, we abolished the dopamine transporter, self-administration still occurs, the whole dopamine hypothesis is wrong. That was a blow, and there were a lot of, at that time we didn't have Twitter, but there were certainly, there was a big discussion at meetings, and there were comments written, it was claimed that is the ultimate blow to the dopamine hypothesis, and we really have to look at this. So, what happened since? Well, the first thing actually is, and that paper was cited in the initial study, is that there was something funny with these dopamine transporter knockout animals. They were actually hyperactive, and that was known before, that was already published. So, those among us who said, well, with these knockouts, genome-wide and during development, who knows, there might be some adaptation. The thing was even worse because there were also other knockouts that were proposed. So here we have the dopamine transporter that shows a typical drug adaptive behavior, this conditioned place preference, that was also present. So that was again against the dopamine hypothesis, but something similar happened with the serotonin transporter. So that was a sort of a question whether that is what exactly is going on here. So then, of course, one could say, well, maybe these monoamine transporters, they're not fully selective for one monoamine. So maybe somehow the serotonin transporter in these mice can still do something when cocaine comes. And cocaine definitely binds to the serotonin transporter. So maybe this is a sort of a additional mechanism that one has to take into account. So what some other people did, therefore, was a double knockout. Right? So now you try to knock out two monoamines, so the dopamine transporter, and, well, sure enough, conditioned place preference did disappear. But, we're not totally satisfied with that because obviously knocking out two of these major monoamine transporter could have yet other adaptations, and there is a third one that cocaine can bind to, so that would have to be examined as well. And actually, the interesting thing was also that this observation here, reported in 2004, looking again in the dopamine transporter mouse, and this time now looking at serotonin uptake inhibitors. And these serotonin uptake inhibitors, which normally are not increasing dopamine, obviously, because they're selective for the serotonin transporter, in these knockouts actually had an effect on dopamine. So this certainly fueled the idea that maybe these monoamine transporters could somehow substitute for each other, and that this could be the explanation why in the artificial situation of knocking out the dopamine transporter, you could still have the reinforcement, which after all was because of the dopamine. And remarkably also is that fluoxetine, which normally does not induce any place preference, in the dopamine transporter knockout actually did cause a place preference. Again, in line with this hypothesis of mutual substitution. But I guess what really sort of nailed the entire explanation was the following experiment here, which was published in 2006 by the group of Howard Gu. And what they did is they refined the genetic manipulation in the mouse. So rather than taking out the dopamine transporter, they provided a knock-in of the dopamine transporter that could still transport dopamine, but has lost its binding site for cocaine. Right? So this is a much more subtle intervention, because all you do is you abolish cocaine, you prevent cocaine from functioning, but you have a situation where the dopamine transporter is almost functional, right? It's not exactly the same, and under these circumstances, they were able to show that you had, in the wild type, of course, after cocaine, an increase in dopamine. That was also the case with amphetamines, but when they used the dopamine transporter knock-in, they totally lost that effect for cocaine, but it was maintained for amphetamine, which molecularly works slightly differently from cocaine. Okay? So I think, when I read that paper, I was, I felt better. And they also showed that under these circumstances, cocaine was absolutely inefficient in driving condition-place preference. So now we have not only a very clear behavioral observation, but we have a molecular explanation for what happened with the dopamine transporter knock-in, and by refining the genetic manipulation, we had the explanation that this is really the most likely target that cocaine uses in order to increase dopamine and eventually use. I mean, I'm showing this here just to sort of convey the message that, well, two things. First, it's not enough to do a gene-behavior correlation, right? Nobody would do this today, but in the late 1990s, that's what we did, right? We had a knockout, we had behavior, we had nothing in between, and we made conclusions that today are simply wrong. So that's the first thing. And the second is, with that, we really have the demonstration that scientific progress is not necessarily linear, right? Between 1998 and 2006, the field went something like that, and there was a lot of uncertainty. I'm not saying that this was lost, because we certainly sharpened our arguments and did many experiments that in the end were extremely helpful, but it was not a straightforward progress in science. And I guess that's often the case, right? I guess those who have been in science for many years would agree with me that, contrary to public perception, not every progress in science is necessarily still valid after some time, and sometimes it's actually throwing the field back more than it actually is contributing. Okay, so we can still use cocaine and dopamine, that's good. But now let's focus on this group here, which is a little bit more complicated to understand, because they work indirectly. That was pretty clear from the beginning, that for opiates, for the club drug gamma-hydroxybutyrate, that was not on the first panel either, the benzodiazepine and cannabis, you cannot evoke a direct effect on the dopamine neurons, because, for example, the opioid receptors, the corresponding opioid receptor responsible for the reinforcement, are not really expressed on the dopamine neurons. Certainly, if you look very carefully, you find one or two dopamine neurons that express mu receptors, but even that could not be the explanation, because these mu receptors coupling to GIOs would inhibit these neurons. So somehow it didn't work. But we knew that there is a strong expression of the mu receptors on the GABA neurons, and they are expressed both presynaptically and postsynaptically. So the idea is that what they do here is they hyperpolarize these neurons, because they do have these Gerg channels. They decrease the release probability, and together they sort of take out this inhibition, and this is the reason why we believe that opiates work through disinhibition. Again, there are other papers in the literature that contest this model. There are totally alternate models based on different experiments, mostly pharmacology, And this, I think, is still not an entirely settled question, which is why the thesis project of Julie is exactly to exploit all the modern tools that we have to reassess that question. But that is going to be for next year's school or the one after that, and I'll tell you whether our predictions will hold true or not. So cannabis works similarly also. We have primarily CB1 receptors on the terminals of the GABA neurons and much less so on the dopamine neurons. So that could also fall into this class of disinhibitory neurons. Okay, so now for the GHB, things are a little bit more complicated because the molecular target of gamma hydroxybutyrate is actually the GABA B receptor. Who has heard of the GABA B receptor? 4? Okay. So this is an equally important receptor as the mu receptors, and it is also GIO coupled. And it was clear from the beginning that there is actually a quite strong expression of GABA B receptors also on the dopamine neurons. So how could it be, or is it possible at all, that there could be a disinhibitory model as well? And it actually turned out to be the case, because what was going on is that, and we could show this experimentally, is when you use GHB at increasing concentrations. So first there's a baseline, then you use a low concentration of GHB followed by a high concentration followed by the GABA B receptor antagonist that is very selective. And at the same time, you record the firing frequency of the dopamine neurons. And what one sees here is that you have an increase in the firing frequency at the low concentration, the cell is shut down at the higher concentration, and everything is restored to baseline with this GABA B receptor antagonist. So the conclusion from this is, yes, indeed, it works through GABA B receptors, and a whole molecular analysis that I'm not going to go into today showed that what was different is that the coupling of the G protein coupled receptor and the effector channel, these GERK channels, is much weaker, actually an order of magnitude weaker in the dopamine neuron compared to the GABA neurons. And that was due to the subunit composition of these GERK channels. So this was another thesis in my lab of Gwen Labweb, and he published 2 papers on this, and that is now a model that is accepted for the addictive properties of gamma hydroxybutyrate. So we have the case for the mu receptors where there is a cell type specific expression of the receptor. We have the case of the GHB where you have a cell type specific expression of the effector channel leading to the disinhibition, which is why we then finally looked into another class of addictive substances, which are the benzodiazepines. So benzodiazepines are prescription drugs that are usually prescribed to induce sleep or reduce anxiety, but it's also clear from the clinical literature that they have not a very strong, but they do have an addiction liability. So the question was whether there could be a disinhibitory phenomenon through these through the benzodiazepines as well. So what is the primary target of the benzodiazepines, the molecular target of the benzos? Which GABA receptors? The GABA A receptors. And what do they do on these GABA A receptors? Huh? Sorry, Yes, so this is the important message here. They are not agonists, so they don't actually activate these receptors. They only amplify the effect of the endogenous ligand, which is GABA, obviously, because they're GABA receptors, right? So how could that lead to a disinhibition? So when we started to investigate this, we realized that there is, again, a cell type specific subunit distribution between the GABA neurons and the dopamine neurons of the ventral tegmental area. So we have here the immunohistochemistry, that is the quantification based on images like that. And what you can see is that the green cells here, the GABA neurons, they only are co-localized with alpha-1, and that's almost never the case with TH. And we did other immunohistochemistry to show that the major subunits of the GABA receptors are the alpha-2, 3 in these dopamine neurons. And then we were able to show that the unitary conductance, so the sort of the size of the minimal current, is actually larger here than here, and that difference would be amplified by the benzodiazepine. So that was our sort of working hypothesis. And in order to experimentally show that this is indeed the case and to link this to behavior, what did we do? Did we use a knockout? No. After what happened to us with the cocaine, we stay away from knockout, so we used the knock-ins to demonstrate that. And the experiment went like that. So first, in a wild type, we did in vivo recordings of the firing of dopamine neurons. And when you put on a fast-acting benzodiazepine, midazolam, you see that the firing frequency, which is relatively low in the beginning, increases, and that can be reversed by the antagonist. Okay, so here we have the effect. And if you only had that, you might even speculate that may be a direct effect on the dopamine neurons. We then did the same experiments in the GABA neurons of the VTA, and here you see the mirroring effect, right? So here you have a decrease in the firing frequency during the presence of the benzodiazepine that is reversed by the antagonist. So how to explain this? So as I said, we took advantage of the alpha-1 knock-in mice because that was our hypothesis that these are expressed selectively on the GABA neurons. And when we did that and looked and did the exact same experiments again in these knock-ins, the lines were flat. So there is no modulation of the firing frequency, neither in dopamine nor in GABA neurons. But since we previously had shown that the alpha-1 is only present in the GABA neurons, the only explanation that made sense was that this is, again, a disinhibition, right? You follow the argument here? Oh, some faces don't seem convinced. Okay, so we have alpha-1 containing GABA-A receptors selectively expressed in the GABA neurons. We put on a benzodiazepine. That explains why we have this inhibition of the firing frequency, because they have a stronger inhibitory drive. And as a consequence, the dopamine neurons that are downstream of the GABA neurons will disinhibit. Okay? Yes?
Did that also abolish the anxiolytic effect?
Well, that's so.
So maybe this benzodiazepines are reinforcing because the absence of anxiety is kind of pleasurable.
Well, let's first see whether there is any behavioral effect whatsoever, right? Because for now, we're just looking at firing frequencies of dopamine neurons. So that could still be totally dissociated from any, So here, what we are doing is a very simple behavioral test, because now we have these knock-ins, so we can actually give them a choice between two bottles. One bottle that contains, in the beginning, water, and the other one that contains water as well. And when we do enough mice, we see that there is no bias, there's no preference, okay? Then we switch and add a little sucrose. We have to do this because benzodiazepines are very bitter. So to mask that bitter taste, we had to resort to that trick to mix this a little bit with sucrose. So if you ever take a benzo and you actually, on your tongue, you'll see they're very bitter. So that allowed us to show that in this situation, we still have no preference. And then we added to the sucrose the midazolam in one, but not in the other, and that led to a strong preference of the relative consumption that was abolished in the alpha-1 knock-in. Okay? So now, of course, that still doesn't show exactly why it is, and the anxiety. So if you postulate that dopamine activation could also modulate anxiety, or if you say the effect actually is totally outside of the VTA, because that's other areas with that design that we also hit. Yes, that is an alternate explanation, which, however, I think is unlikely because there are other knock-ins that have been shown to mediate the anxiolytic effect. So just to show you again the power of these knock-in animals that initially are not very interesting unless you actually want to study pharmacology. And of course, the people who have generated these mice in Zurich, Uwe Rudolph, their purpose was to do precisely that, right? To dissociate different effects of benzodiazepines with a cell type specific pharmacology. And that's now, as you can see, I mean, the knock-ins, they were published 15 years ago, and still we do not have cell type specific or subunit specific benzodiazepines. So while in basic research that's very convincing and extremely well published, so their initial report of these mice in Science sparked a lot of hope that we could actually go there, but for reasons that I'll be happy to discuss, this did not pan out, and in 2016 we are still left with benzodiazepines that are totally unselective, with the exception of, there's one benzodiazepine that is subunit specific, the zolpidem, right, or Ambien in the US, or Stilnox in Europe, and that was actually introduced with the promise that it would be less addictive. Right? And now we identify the alpha-1 subunit, which is the specific target of this zolpidem, as the culprit for the addiction process. So when we published that paper, within actually a week, I got a letter from a lawyer from a big pharmaceutical company that was marketing zolpidem. I'm not going to give you the name, you can look it up. And they said, we're going to publish a matter arising in Nature to show that everything you say is wrong, because we know and we've marketed this molecule because it's less addictive. And, the evidence that they had was not very convincing, and therefore the matter arising was never even published. But just to show you that sometimes what you do has at least sparks the interest of others. Okay, so now I guess this is where we are now. I don't think it is fully established exactly, but I guess we've done some of our homework to show that there is this convergence onto the dopamine neurons by the different addictive drugs. And I think that was important, at least for me, to have the confidence that we should study this system if we are interested in addiction. Yes?
May I have a question? What about the endogenous substances? Do they lead to increase in the dopamine?
So what endogenous?
Like endorphin, like endocannabinoids?
Sure, that's well possible. I mean, this system obviously, and that would just my next slide, it has a physiological role. And there are situations where these cells are stimulated, and that could be through different means. It could be through means of increased excitatory drive, decreased inhibitory drive, modulation through endocannabinoids or enkephalins and so forth. Yeah.
But
so
they usually don't lead to addiction.
To addiction. Well, that is the question we will have to discuss. I mean, if you have behavior X that drives that input or that release of endocannabinoids, maybe this is a behavior that has a certain addiction liability. So if you could show that in situation of gambling, you activate that system, maybe you have a unifying hypothesis that would extend beyond the pharmacological one. But of course, there is some evidence, particularly from human imaging, but of course, to understand this at the molecular level, it's a little bit more tricky. We'll discuss also a little bit in the paper our approach, but to have a mouse gamble I'm not saying it's impossible, but and to have them so much involved that they eventually get addicted, that is tricky. Because, for example, the lifetime prevalence for human pathological gambling is how much? Is it lower or higher than cocaine? Higher. No, actually, it's estimated to be around 1%. So it is much lower than cocaine. But still, if you have 100 people going to the casino, one who gets a pathological gambler, this is a problem. But so behavioral addictions are typically less so, but still there's, and of course, the direct comparison is extremely difficult because the methodology cannot be the same. Yeah. Okay, so obviously, as I said, this system is not here just to be the target of addictive drugs. It has a physiological role. And we could fill an entire week of discussions to come to conclusion what is the role of the mesolimbic dopamine system. So I'm not going to go there. I'm going to give you a highly biased, just a very tunnel view of what it certainly also does. I'm not saying this is exclusive. And this is derived from these seminal experiments that were done in Switzerland by Wolfram Schultz and published in 1997 and cited more than 4,000 times by now. So what did Wolfram do in macaques? He recorded the activity of dopamine neurons without distinction between VTA and Nigra, but that's a side note. But what you can see here is that if you put together several observations where you have single units that you record from, you can come up with these plots here where the vertical ticks here indicate the number of action potentials. And so when these macaques received a drop of juice, which was a reward for them, you see that there is a slight activation of these dopamine neurons. So, first conclusion from this experiment, dopamine neurons in the VTA, or in the midbrain for that matter, code for the reward. Okay? Well, it did not seem to be the case always, because that experiment only worked if the reward occurred without anticipation, so by surprise. So contrary to that, when you actually give the monkey a warning, so you have a sort of a conditioned stimulus, this image here, that tells the monkey that 1.5 seconds later they will get the reward, what do you see now? You see that there is no activation of the dopamine neurons at the moment of the delivery of the reward, but there is an activation at the moment of the presentation of the conditioned stimulus, which now comes without anticipation. So how do you have to refine your hypothesis from saying that they code for reward? Huh? Unexpected reward. So an reward that comes by error, right? So here, Wolfram Schultz introduced this idea of prediction error. So here, the idea is that the monkey does not predict the reward, receives a reward, and so there is a prediction error, right? Here, the animal does not predict the conditioned stimulus that comes by surprise, so there is an activation. But here, the reward is entirely predicted, which is why there is no reaction. So if you have that, what experiment did they have to do to confirm their hypothesis? Yes? Exactly. This is a very unfair experiment, right? You promise a reward and you do not deliver. And the prediction would be that under these circumstances, the cells shut down. And this is exactly what they saw. Okay, so you have the prediction error in both directions. The positive prediction error, this, and the negative prediction error here. Okay? So that was, and actually, it seems like an abstract thing, but we're all very sensitive to that, right? If I now gave you $5,000, this is not what you would expect from a course like that, and you would probably sign up again next year. You know, it would change your behavior, and you would do everything possible to have it again. If you get a sum every month, like your salary, because people promise that to you, doesn't do much, right? I mean, you don't even notice when you get the salary until the moment where they have promised you the salary, but they don't pay you, and then you react again, right? So prediction error is in a daily life. I mean, it really is an important entity. Okay, so why am I telling you this? Because it may have an impact on a model on how we conceptualize the initiation of drug addiction. And this is actually very nicely summarized by a recent review from Patricia, where she puts this in the context of a learning paradigm. Okay, so here you have the exact same graphical representation of the experiment that I just showed you, right? In the beginning, you have the cue presentation that does not signal the reward yet, so therefore the reward comes by surprise, and that activates the dopamine neurons. With time and what is called here moderate training, you actually see that you start to have a reaction, a peak that emerges at the moment of the CS presentation. And then when there is extended training, the animal is totally used to that. There's zero reaction to the reward, and all the dopamine neurons are activated only at the moment of the CS. So the important thing here to realize is that there is a sort of a devaluation of the reward. So at the moment of the reward, there is no activation of the system.
But why is not abolished in the stimulus?
Sorry?
Why is not abolished in the stimulus when the reward is not activating any impulses? Why the stimulus would activate?
Well, because now the whole thing has been transferred to the conditioned stimulus.
But it must get used to the conditioned stimulus.
Well, yeah, of course. I mean, if you do this over and over again, such that the CS becomes predictable, you start to lose that as well. So that has to be timed such that the CS cannot be predicted. That's important. Yeah, this is a very good remark. Yeah. But now, in the situation of addictive drugs, what happens is that regardless of where actually you are in the training, there is always a very strong activation by the sheer pharmacological power of these substances. And particularly in the situation where you should not have an activation at the moment of the reward. Okay?
But they show this also in animals in panel B, were they tested for addiction, or were just all the animals were responding the same way?
Well, no, this is the initial phase here. So for a moment, we don't, you know, again, the first slide was important to show that we have different steps through which addiction develops. And here, you cannot say yet that the animal is addicted. And I will actually even argue that even in a fully addicted animal, there is no difference in the dopamine signaling. You know, I mean, I'm probably not 100% right, but we have different views on that in the field, and I'll give you my arguments why I think that the differences in the actual dopamine signaling between addicted and non-addicted animal is not really the relevant parameter to study. But what you can see here is the Q value, which obviously increases as these firing frequencies increases here, is sort of leveling out because of that devaluation situation. And that is different here because with the drug, that devaluation is not possible. It's overridden by the pharmacological power. Okay, is that a fair summary of your figure? Good. Yeah, no, it's a great article. You should read it. Okay, so therefore, beyond that increase in mesolimbic dopamine, we have to find what is the correlate. So the commonality is this convergence onto the dopamine, but then after that, something needs to be driven by that dopamine, and because of the context and the whole theoretical ideas on learning and how dopamine could be a learning signal, synaptic plasticity was an obvious candidate. Okay, so the idea again, drug-evoked synaptic plasticity eventually leading to the changes. So now, in that second part that is going to start now, we're going to talk about this drug-evoked synaptic plasticity. And the first description of such a drug-evoked synaptic plasticity is this paper here in Nature in 2001. And what is shown here is an ex vivo experiment. So one single injection of cocaine, you let the animal live for 24 hours, it is then killed, and slices of the ventral tegmental area are produced. And in these slices, you then record synaptic transmission, and it's the glutamatergic afferents onto these dopamine neurons. And what you can see is they have two components. They have an NMDA component, here measured at plus 40. It is relatively slow and relatively large. And in the naive, they have at plus 40 a relatively small AMPA component. Okay? That then is the same after a control injection of NaCl. That should not do anything. But what you see here in the cocaine condition is that things change. It seems as if the NMDA component gets smaller and the AMPA component gets bigger. But of course, there is one parameter that escapes this experiment, which does not allow you to argue in these absolute values, which is in a slice. Who does slice physiology? Yes, so what if I were to say from these example traces, and I can show you many more from my animals, I say that in absolute values, the NMDA went down and the AMPA went up. What's your argument?
I mean, you're taking a ratio, so I would say it's...
No, I'm not taking, I'm just don't want to take a ratio.
Yeah.
You say I should take a ratio.
I'm saying you should take raw occurrence or raw mediators of occurrence. Is that what you're saying?
Well, I'm saying, I would like to make the argument that in absolute values this changes, but you're saying we should make a ratio, and you're right. And why are you right?
Well, the idea is because MDA receptor composition doesn't change usually, but receptors do. It's an indication
Well, not so sure that this is exactly, I mean, there's one parameter that in this experiment can totally screw you up. The size of a response depends on, obviously, how strong the current is at one receptor, but it also depends on the number of synapses that you stimulate, right? And between two slices, because of the slicing procedure and so forth, there's a consensus in the field that you cannot compare this in absolute values, okay? Which is why, for many years, all what people did in the field is what to do the AMPA and MDA ratio, okay? So this is the precise reason, because there's this one parameter, which is how many synapses you look at, how many fibers you stimulate, that cannot be controlled for, and therefore we prefer to express this as a ratio, and then we do some experiments to show that one of the terms is not changing and infer that the other is increasing. And this is exactly what was done in that paper. They said, you know, we have evidence that the NMDA component does not change, therefore the increase of AMPA and MDA ratio must be because of the insertion of AMPA receptors. All the observations are right, but probably interpretation has to be revisited. And the reason why it has to be revisited, you already alluded to, is that the AMPA subunit composition changes. And that was then shown here in this experiment here through using, you know, one example of each of the three groups of addictive substances. And as you can see, when you isolate the AMPA component, so these fast glutamate receptors, and you record them at plus 40, at 0, and minus 70, you see that in the situation of the cocaine, when these currents are normalized to minus 70, you have a clear deficit at positive potentials. And therefore, the current voltage relationship, which originally is linear, changes and becomes inwardly rectifying. Okay? So obviously, we'll discuss what that means. But first, we really want to be sure whether the whole thing has something to do with drug addiction or the initial effect of cocaine onto the dopamine system. So what is it that you would do to show that this is really, because then again, we're working with pharmacology, and pharmacology inherently is non-specific. So how would you show that these synaptic changes, which are here for cocaine, but also for other addictive substances, may represent a first trace that the drugs leave behind once they've been cleared from the system? Yeah.
You do the same experiment, but
Huh? Knockouts. No! The knock-in, right? You want to do it again? Now that you've gone for 10 years, we were totally, now here in 2010, so now we know how to do it. And so the obvious experiment to do is to repeat these experiments with the dopamine transporter knock-in that cannot bind cocaine, right? And the prediction is, if this is really downstream of the increase of dopamine, then we should no longer have these synaptic adaptations, right? Okay, so first, we actually did in vivo recordings to show that under normal circumstances, when you give cocaine, the firing frequency goes down. That's surprising, no? I mean, we were not the first to report this, but so this seems to be the effect on the firing frequency of cocaine in the in vivo situation. How is that possible? Because we argued that drugs increase the firing frequency of dopamine neurons, and here we clearly see that there is a decrease at the moment we inject the cocaine. It's a difficult thing to understand, but what's going on is this is not the effect of cocaine on dopamine, it's the downstream adaptation of the cell once the dopamine massively increases. Because these dopamine cells have D2 receptors, which then get activate the GERGs, and that hyperpolarizes the cell and decreases the firing frequency. But despite that decrease of firing frequency, the overall dopamine actually goes up. So this is it's a difficult thing to understand, but that's how nature works. What can I do?
So the dopamine
right?
went up, but then
these cells release dopamine here. You come with your cocaine, you block the reuptake, dopamine goes up, it does his spiel on the reinforcement, but it also has an auto-inhibition of the dopamine neurons through the D2 receptors. Exactly, yeah. But the net effect is still a massive increase of dopamine, okay? But importantly, in the knock-in, this is gone. So here we have a cellular demonstration that the knock-in is really efficient, okay? And so that is shown here in this group data. So with that validation of the mouse, if you get a mouse from another lab sent to us and you rebreed and everything, there's so many things that can go wrong. It really is important that you validate that it does what it's supposed to do. And then we did this experiment here that you now know. So there is the saline with the dopamine knock-in, the wild type. The wild type that shows the strong rectification, and with the cocaine, the dopamine transporter knock-in no longer shows the rectification, and here's the group data. Okay? So this is really, then when we saw that, we were really interested, because now we have not only an addiction trace that outlasts the presence of the drugs, that is here with all addictive substances tested so far. But we also can show that, at least for the situation of the cocaine, it is downstream of the dopamine. Okay? Okay, so now what does it mean that this rectification is here? So the rectification indicates a specific subunit composition of the AMPA receptors. So AMPA receptors, this is the old nomenclature, so it should be GLU-A1 and GLU-A2, so they're heteromeric channels. And this GLU-A2 has a specific property, that is, it is edited such that there is a large arginine residue in the pore region, and because this is so big and positively charged, calcium cannot enter the cell, and the single unit conductance for sodium is decreased. And as a consequence, we have a linear current voltage relationship. So in AMPA receptors that do not contain GLU-A2, we have a glutamine in the pore region that is much smaller. So now sodium can flow in much more readily, and at the same time we have a permeability for calcium. And because at that point polyamines that are normally present in the cell can access a specific inhibition site at positive potential, we have this inward rectification, which simply means that at symmetrical potential, the outward current here is smaller than the inward current. So applied to our situation, what does that mean? It means that we're going from a VTA, where dopamine neurons have the GLU-A2 initially, to a situation where we have AMPA receptors that lack GLU-A2. And that strengthens the input because the sodium conductance is higher, and at the same time MACE is permeable to calcium, and we'll see that this has consequences. Okay, so one more element to show you that this is really through dopamine that this occurs is obviously to do the converse. So we showed you the necessity of the dopamine transporter and the effect on the dopamine neurons. Now we can also show you, using the modern tools of optogenetic, that this is actually sufficient. And the experiment you want to do is, if you wanted to show that the activation of the dopamine neurons is sufficient to induce this form of plasticity, huh? What? Well, you don't even have to self-stimulate, but you have to find a way to stimulate the dopamine neurons and thus mimic, for example, what nicotine does, right? And how do you do this? With optogenetics. So now we use the DAT-3 mouse. We inject virus into the VTA that will infect all neurons, but recombination can only occur in those neurons that express the dopamine promoter because of the Cree element. And then we replace the injection cannula with a light guide, and then we can do, for example, the experiment like here, where we have for 2 hours a burst firing pattern that mimics what these dopamine neurons can do best when they want to release massively dopamine, and then we do, after 24 hours, the slice recording, and you see that we can induce the rectification simply by strong stimulation of the dopamine neurons. Okay, so we've shown necessity and sufficiency in this. And we can even show that a local infusion of the D1 receptor antagonist in the VTA prevents that plasticity from happening, confirming that an induction mechanism that had previously been postulated. Okay, so the AMPA-NMDA ratio changes because of the change in the AMPA receptors. But that's not the entire story, and that took us almost 10 years because we realized that the NMDA receptor subunit composition also changes. So here I show you a typical NMDA current voltage relationship that is inhibited at -80, then gets some positive, and then is outward rectifying. And after cocaine, that actually is different. And so we have not only of course, I mean, there was an entire story. I'm going to make this a little shorter. And we not only have a change in the AMPA receptors, but we also have a change in the NMDA receptors. And that decreases the absolute magnitude of the NMDA current. So the initial interpretation that we dismissed because we were very strict about the number of synapses in the slice preparation was actually true. There is both an increase in AMPA and a decrease in NMDA. Okay? And that is interesting because it leads from a situation where calcium can flow into these cells only by depolarization in the naive state through the canonical NMDA receptors, and then is replaced by this double exchange of AMPA and NMDA receptors, where now the calcium can come in through AMPA receptors and actually much less so through these new NMDA receptors that are very inefficient in conducting calcium. So why could that be interesting? Because what does calcium do that comes through NMDA receptors? Huh? Yeah, you have to depolarize, then calcium comes in, and downstream of that calcium, huh? Well, yeah, synaptic plasticity. I mean, that's what NMDA receptors were invented for, right? To bring calcium into the cell and then trigger LTP, for example. Okay? So here, to get the calcium in, it's good to depolarize the cell. Here, to get the calcium in, what do you have to do? You actually have to hyperpolarize. The more you hyperpolarize, because of the inward rectification of the AMPA receptors, the more calcium you bring in. Okay? So based on this reflection, Manuel, who actually is going to come tomorrow or so, did the following prediction. He said, if I want to induce plasticity in these cells, yes, I have to depolarize. I have to use a canonical LTP protocol. However, if I want to induce LTP here, I have to hyperpolarize this. And this is exactly what he saw. So here, he uses a sort of a spike timing dependent form of plasticity. If you have in the naive, you have a dysynchronous activity of these, you do not get any changes. Now you depolarize synchronously the postsynaptic when you activate the presynaptic, so the activity arrives on a depolarized neuron and you have the potentiation. In the situation where you have this synchronous release but after cocaine expression, since these NMDA receptors that can conduct while being depolarized are no longer present, you lose the ability, that's all a little fast. You lose the ability to you lose the ability to induce LTP. Okay? Maybe I have to. Okay, here. Okay. Yes. Now he no longer could get LTP. Okay? And you can rescue this by imposing a protocol where the postsynaptic cell is hyperpolarized each time the action potential is derived in the first one. Okay? So in other words, what cocaine does is it changes the rules of engagement. It changes from a classical NMDA receptor-dependent form of plasticity to a calcium-permeable AMPA receptor-dependent form of plasticity where you have a pairing between a hyperpolarized neuron and an active neuron upstream. Okay? Okay, this is again summarized here, or in other words, what we could say is that these initial changes are not necessarily something that immediately drive the way the system changes, but it is a sort of it's a metaplasticity, right? It's a plasticity that allows for new forms of plasticity. Okay? Okay, so with that understanding, we then tried to see whether we could correlate this to behavior. And that was actually many years ago, so our first attempt was to use actually a knockout. But at least, in our defense, we didn't use a sort of a genome-wide knockout that was already present during development. We at least used an inducible and cell type specific knockout. So that is, here, by crossing a CRE-ERT2 containing mouse with an NR1 floxed, you obtain a knockout only in the dopamine neuron when these animals are exposed to tamoxifen. Okay? So the experiment went like that. You had this tamoxifen, and then there was a slice preparation several days later, and we could show that this was enough to abolish the NMDA current. Right? So here in the CRE+, no NMDA current. This is this slow current that we saw before. It's now only AMPA current that is present. The CRE negative ones still have the NMDA current, and the neighboring GABA neurons also have NMDA currents. So it's cell type specific, and it occurs after a number of injections and the delay that allows for the downregulation. Okay? So now with that, we could now test whether the drug-evoked synaptic plasticity was gone. And so indeed, what you can see here in the wild type, we have the rectification. So you're now familiar with these plots here, and that was no longer present here, okay? And this is the group data. The rectification index was gone. So with that validation of the system, we felt that we had all the tools needed to look for drug-adaptive behavior. And since we knew that the induction of this took about a day, and it lasted for about a week, the obvious attempt was to use drug-adaptive behavior that had a similar time course, okay? And so we looked, for example, condition-place preference, right? This is the test that we already mentioned several times, where you have two compartments, one of which is paired with cocaine, and then you look whether that leaves a trace in an animal. And you can actually get this already after 24 hours. And no, we did not have the behavioral correlate at the time where we had the plasticity, because that CPP that also started about a day and lasted a week, that was absolutely normal. And so was extinction, and it's only reinstatement, which was weeks later, where we saw a difference. So this was a sort of a very difficult moment in the whole lab, because we realized that we had a very specific handle on this plasticity, but the behavioral repercussions occurred much later. There was a total temporal disconnect. And so we didn't really know what to do until we realized that maybe this has something to do with a hierarchical organization of synaptic plasticity. So owing to the fact that we actually are not really inducing plasticity, but metaplasticity, the idea was that maybe what we do here will only have repercussion if it's relayed to the upstream targets, such as the nucleus accumbens. Okay, and so we're going to leave the VTA and going to start working in the accumbens after the coffee break.